Hi, Bob.
Hey, Jim, how are you doing?
Good. How are you?
Doing all right. It's kind of eerie up here. Everybody's cleared out. Normally it's light because of COVID, but whenever they shut down the institution, it gets really weird.
Yeah, I'll bet. So how are things? I mean, obviously I can see you got some rain behind you, but does it still look pretty?
Yeah, the nasty stuff is about to hit now. It's that long line that's sweeping across the southeast, and I just set the weather forecasters are just, when they get all Twitter pated and excited about this, it's going to be bad. So, when they've been talking about for 3 days now. So like, okay, they saw something. So anyway, but we're good. Like I said, I'm at the office, and so I can't imagine we would lose power here. That's my only concern about trying to do something from home, although I have a better setup for Zoom there than I do here. But that's all right. And I forgot, where are you located?
Pittsburgh.
Okay. For some reason, I was thinking you were in the Raleigh area or DC area. I don't know what, but yeah.
So let me see. Let me share my screen and see how. Let me know if you can see the intro slide here.
Yes.
Okay.
Yep.
All right, great. So do you want to try to go ahead and share your?
Yeah, let's make sure that I have to wait till you quit sharing. How's that?
Excellent. Yeah. Fantastic.
Great. So you want me to leave it on or?
Well, you can leave it up. I will.
I need.
Yeah, why don't you let me share? I'll share like my my screen there, and
Okay.
Yeah.
Sounds good. All right, so I'm going to turn off email so I don't get any beeps, which come way too frequently. do my best to not go over.
Oh, that's fine.
I'm notorious, and about half of this is all new data that hasn't been shared. So you know, I tend to run on at times and backtrack, and it's unfortunate because it's not going to be as smooth as I want. But I really like sharing new data.
Oh, well, that's fantastic. We like to hear new data. So we'll probably start maybe, you know, like a couple minutes late or something, just to make sure that people have a chance to log in. So what's everybody do when they shut down the university now? Is that just everybody take the day off, or does everybody work from home?
Yeah, that's kind of what it is. You know, they've started doing this more. Let's say the first 10 years I was here, they never did that. But then we had that disaster where the snow and ice froze and people were stuck up here for 2 days. And they then amended their protocols for, you know, paying attention to the weatherman and letting people go when weather gets really bad. And it's also for people to get home and get their kids from school, make sure they're safe, and those sorts of things. You know, it's just unfortunate. I mean, you know, this part of the country gets really bad weather at times, and I mean, Pittsburgh gets a lot of snow. I know that, but well, you guys are prepared for it.
Yeah, well, we got a good bit of snow this year, although in a few years past, doesn't seem to have been as bad. And this year, it just kind of like somebody threw a switch at the beginning of March, and it just stopped snowing, and it hasn't snowed since.
Yeah, so they're in very strange weather patterns for right now.
All right, I'm going to start letting everybody in here. It looks like we've got a waiting room of 40 people. Okay. So
hello.
Afternoon.
Hello. Thanks for speaking to us. Hello, everyone else.
Good afternoon.
Yeah. Good afternoon. Hi, everybody.
Hi.
We'll give more people a little bit extra time here to log in before we start.
I don't know if anybody celebrates St. Patrick's Day, but happy St. Patrick's Day, everybody here from Ireland.
Oh, indeed.
Happy St. Patty's Day.
Happy St. Patty's Day, yes.
My son is celebrating in daycare. They are all very green today.
Stay away from the green beer.
At least until after the meeting, the seminar.
Nobody drinks green beer in Ireland. It's a complete myth.
The worst is when they stain the Chicago River green every St. Patty's Day in Chicago.
They still do that?
I don't know if they still do it, but they used to. It's kind of crazy.
Yeah, they did it again. I saw the photos. It's you know, really, really hideous green too. It could be GFP.
Let's hope green dye #1 is not as bad as red dye #1.
Yeah.
Okay, well, I think we've got quite a few people participating who've logged in, so we might as well get started here. I want to thank everybody for attending our second seminar for the Gilbert Family Foundation NF1 seminar series. A couple of virtual seminar guidelines. Not everybody has seen these before, but again, please mute your microphones until the question and answer portion of the talk. If you have any questions, you can type them into the chat box. And if you have any issues with Zoom, you can private message Caroline, and she can usually take care of that for you. So I want to welcome today's seminar speaker, Robert Kesterson, professor of genetics at University of Alabama at Birmingham. Bob completed his undergraduate studies in chemistry at the Hendrix College and received his PhD in cell biology at Baylor College of Medicine. He performed his postdoctoral training in Roger Cohn's lab at the Ballum Institute in Portland, Oregon, where he was focusing at that time on melanocortin receptors, which had just been cloned in Roger's lab. He joined the faculty at Vanderbilt University in 1997 and was recruited to UAB in 2004. Bob is currently the director of the UAB Transgenic and Genetically Engineered Models Facility at UAB, and the director of a U54 pilot project for precision animal modeling. His lab uses genetic mouse models, rat models as well, for numerous physiological and behavioral processes, looking at a number of different disease states, but most recently he's been interested and he's been focusing on neurofibromatosis, thankfully for us. So without further ado, please welcome Bob. And Bob, let me get out of here so you can start sharing your screen.
Great. Thanks so much, Jim. And thank you for the kind introduction. And I really appreciate the opportunity to talk to everyone today. And because our group has been creating some unique animal models in the last decade. And what I hope to do today is kind of go through the history of what we've been doing with these genetically modified animals, and tell you, do that briefly, and then tell you about some of the unique models we've been creating at UAB, and touch on our in vitro system for analyzing structure function of various patient mutations, and really describe the genetic models for how we go about examining loss of heterozygosity. And this builds upon what many people have done in the past. And then we have some couple of new models that are particularly, I think, powerful to more quickly assess loss of NF1 function. And so I'll be talking about, and then lastly, I'll touch on some of our nanoparticle project that's funded by the Gilbert family, and some of the challenges going forward for gene editing. So everyone has seen a slide like this if you're in the NF1 field, so I'm not going to spend too much time here, other than saying that there's a pleiotropic effect of loss of NF1, and we see this in patients, and the patients, it's a large gene. There's numerous mutations, and there's no real hotspots. And so, and what we do know about NF1 is that primarily affects the peripheral nerves and spinal cord. There's learning deficits in the children, and a lot of people are focused on the tumor growth and particularly concerns about peripheral metastatic disease, which there's real no treatment. So I borrowed this slide from David Gutman from one of his articles, and I think does a really good job of talking about or demonstrating the loss of heterozygosity that really drives neurofibroma formation, and in particular, the fact that it's really potent in the context of a heterozygous background. So if you already have one mutant allele, then the loss of NF1, in particular in Schwann cells, helps drive the formation of neurofibromas. And again, this diagram captures a lot of work of people that have done amazing things. Please forgive me if I don't mention your name or your mentor's name, but I think Wade Clapp, Nancy Gratner, and Louis Parada, and built on stuff that Copeland and Jenkins and Tyler Jacks first made the knockout. So there's been a lot of work that's been done here. David Gutman, of course, has done a lot with the optic glioma model. So what I want to do is say this is the loss of heterozygosity. And so that's the context of what we need to try to create in animals. And the reason we decided to focus on this is when Bruce Korf recruited me to the field, and it's been 8 to 10 years ago now, and Bruce was really interested in us developing something new so that we could look at mutation-guided therapies. So, some outstanding work has been done with RAS pathway inhibitors. We now know we have a clinical approved therapeutic for NF1, which is great. But we also have a need for some mutation-guided therapies that could augment some of the things that have been recently developed. And so when we look at the mutation spectrum for NF1, we see that NF1 is represented by numerous types of mutations that any gene would have. There happens to be a disproportionate number of nonsense mutations in NF1 gene, about twice as many as you would expect. Typically, most genes have about 10% of the mutations or 10 to 12% are nonsense mutations. So we have a lot of nonsense alleles. And so I will touch on these different animal models and what we've tried to do to create, I don't know what's the function here, back it up. So to create our different mouse and rat models at UAB. So again, I mentioned that historically there's been an awful lot done with the original knockout of NF1 in mice. I think there's a major disappointment in that the heterozygous NF1 mouse did not phenocopy what we see in patients. That is, heterozygous animals are for the most part relatively normal. They don't get, if they get a second hit, they don't really produce neurofibromas. And it wasn't until a conditional knockout model was created and then started crossing these conditional knockouts with the cell type specific promoters to then drive that loss of heterozygosity to really produce a phenotype. So we started focusing on premature stop mutations or nonsense mutations, a couple of those. And then the other type of mutation we wanted to focus on were missense mutations that were associated with clinically a subgroup of NF1 patients, so the patients who have spinal NF. And the spinal NF patients, Bruce, this is a slide that Bruce Korf shared with me. And so you see this patient on the left, so you can see that there are maybe some cutaneous tumors under the skin, so a few, but really the major issue here are these large plexiform tumors. On the right here, you can see this is the spinal cord, and you have these massive tumors emerging from the nerve roots. This is a sciatic nerve of this particular patient, huge tumor burden all along the sciatic nerve. So these are really concerned, these large plexiform tumors, and of course, many of those go on over the lifetime to be peripheral metastatic tumors, and somewhere around 8 to 10% of the cases. So we started creating patient-specific models in order for us to study this. And so what I'm going to do is introduce you to the schematic of the different, the NF1 gene, some of the different mouse mutations and rat mutations we've made. In fact, here is a table that is fairly up to date with almost all of the animals that we have completed so far with different mouse-specific, patient-specific mutations. Many of these are a couple of nonsense alleles, some are some frame-shift alleles, some are missense mutations. And so I'll tell you about a couple of examples of those. And so the bigger question is, well, what do you do once you have a patient-specific mutation? Why are you bothering to do this? And what is the system that you can, or the context that you can put these animals in order to study neurofibroma development and to have some sort of treatment or intervention? So this is a nice cartoon of the different models that we're currently employing. And so we'll do things like making stem cells from or ES cells or primary cell lines from heterozygous animals. Normally, most pathogenic mutations for NF1 are embryonic lethal. And so you cannot make a homozygous animal. In order to study a mutant allele, you need to put it in the right genetic context. So probably one of the traditional, or probably one of the best models pioneered by Nancy Ratner's group and others, was using the desert hedgehog tree to specifically knock out a floxed allele in the Schwann cell precursor lineage. And in that case, you get paraspinal plexiform tumors that normally arise around 4 to 5, 6 months of age, up to 1 year. And so this is a very robust plexiform tumor model. Some of the newer models, recently Lou Lei's group has published a HOXB7 model, which also produces these paraspinal plexiforms, but also has the added benefit of producing cutaneous tumors. David Gutman's lab has been using the GFAP-CRE to study optic gliomas, and these arise in 4 to 8 months. And then there's a couple of new models that I'll go into details about that our lab has developed using either a CAGS-CRE or a GABAergic specific knockout of NF1. So let's start with the nonsense models. So this cartoon kind of describes, well, why would you want to make a nonsense model, and what can you do about a premature stop mutation? And if you have a premature stop, one of the things, you obviously get a truncated protein, and you get the wrong amino acids potentially added to the growing polypeptide chain. Well, actually, you get a termination early. But if you could somehow suppress this mutation, so the treatment therapy called nonsense suppression therapy, with using aminoglycosides, you could get read-through. And in this case, if you could get a different amino acid inserted into the growing polypeptide, you could produce a missense protein that could be functional, or it might not be functional. And so really, this is work that came out of some work I had been doing with David Bedwell, and Kim Keeling at UAB. So when I first moved to UAB, the first mouse model I made was a Hurler's mouse for David's group. And David has been studying these nonsense mutations and using nonsense suppression therapies for both the Hurler syndrome and also cystic fibrosis, which was another model we made. So we've now made 3 different nonsense models at UAB. And the key here is that this is a proven therapy, at least for some diseases. You can get read-through, and there's a drug, Adalurin, that's been approved in Europe for treating Duchenne's muscular dystrophy. And so if you get some partial activity, particularly if it's an enzyme or some sort of enzymatic activity, you could get restoration of function of a gene. And for NF1, that made sense to us in that it would potentially restore partial function to suppress the elevated RAS activity. So Min Chen in my lab first made, using embryonic stem cells, this premature stop mutation. This is a patient-specific mutation. Ludwig Messiaen had shared this mutant allele with us some time ago. We created this particular mutation, and then we combined it with a different conditional knockout allele. The other allele that was available was the exon 31 and 32, but we used a new one created by Sanger that flanks exon 4, and we combined that with the desert hedgehog CRE. We get a model as you would expect, and this looks very much like the work coming out of the Ratner laboratory, in that over time you get paraspinal tumors that compress the spinal cord. Here you can see this neurofibroma here and here, and it's compressing on the spinal cord. And the mice start showing hind limb paralysis. They'll start limping a little when these tumors start growing and pinching the nerve. And the other thing you can see is that you see swelling of the sciatic nerve. And if you look by EM, you can really see that there's demyelination of the major nerve groups and nerves all along the spinal cord with this particular model. So that sets up a potential intervention therapy in which if we were to combine this allele, we could try a treatment protocol in which you could allow tumors to develop and then treat them with nonsense suppression drugs and find out could you then suppress the growth of those drugs, or a prevention type protocol which you administer drugs and see if you can prevent the development of the tumors. Now, these are pretty intensive experiments given the frequency of the tumors. This takes large cohorts to get workable data. We did an early pilot experiment with a drug combination that showed hints of working with the prevention, not so much once tumors are developed. And then we've since learned that one of the problems with this original knockout model that we made is that this particular allele is not really sensitive to nonsense suppression therapy. And working with David's group, and David has one of the Gilbert Family Foundation grants, we now know that this allele is not particularly good at read-through drugs. And so now we've moved to a different nonsense allele, which does show much greater activity and sensitivity to those drugs. So I would expect looking forward that David's group would be giving a talk or presentation on this, really showing how we could use the in vitro data to help get better drugs to then treat our different animal models. And I'll come back later to what would be a better way of testing this. So our early missense mutation model that we made, we decided to make one as a very pathogenic mutation. And that's this, hang on a second, I need to move some things off of my screen here. I don't need to see everybody. Let me go back. So this pathogenic mutation at position around 848 as this glycine to arginine, and Ludwin's group has published that this particular region has a fair number of collectively of pathogenic mutations. And so this is part of the spinal NF that she had noted in a couple of the patients or in cohorts. Actually, independent families had the same mutation, both had spinal NF. So we created a mouse model. Again, Min Chen did this in the exon 21 of the mouse. And to our surprise, we could create heterozygous animals and homozygous animals with this mutation in that it didn't appear to be pathogenic at all. And in fact, if you look at the survival curves of these animals, the homozygous animals for this particular missense mutation appear to be relatively normal and survive just fine, nearly as long as the wild types. When we look carefully at the brains, we also looked at MEFs that were isolated from the cells. One of the things we noted is that there was less protein expressed though. So in the homozygous animals, there was much less protein. In fact, that was almost to the level that a heterozygous null animal would be. And along with that, you had elevated phospho-ERK levels. So there is a consequence of this particular mutation in vivo. It's just in the mouse seems to tolerate it just fine. And many of you know from Frank McCormick's work and Dominic Esposito, they've been very interested in studying this mutation. And they found that if you make the same mutation in a human cDNA system, that protein is really unstable. So we do think that the stability is a major issue with this mutation, but in the context of a mouse NF1 gene, it's okay. So it doesn't seem to have any major problems. So creating those mice really took us a long time, and much like today here in UAB where we have tornadoes and everything coming through, it was kind of dark days. But then, and I stole this slide from Tyler Jackson, I heard him present it, because then what came to us as the sun really started shining on us was CRISPR-Cas9 came along. And as many of you know, this has just revolutionized the way that we do things. And then the other key thing that came along for us is that we were able to isolate a full-length mouse cDNA. And I know in the field had really struggled for quite some time trying to piece together a full-length human cDNA. It's extremely unstable. It rearranges every time you try to grow it in bacteria. We've experienced some of the same thing. We're still working on putting together a mini gene for NF1, but it has been problematic getting a good cDNA. And so the reason these two kind of changes for us were important was with CRISPR, whereas it took us approximately 1.5 to 2 years to make those first animal models, we can now make a knockout or knock-in model much more quickly using CRISPR-Cas9. And in fact, most of our models now, with a few exceptions, most of them we have within 9 months to a year, but some of them can take much longer, particularly for lethal genes in which using CRISPR to create an animal, you end up hitting both functional alleles. So I don't want to spend a whole lot of time on this, but really all the CRISPR does is introduce a double-stranded break. And normally you end up with getting indels through non-homologous end joining, so you get extra alleles, or that's the way the animal will repair the introduced double-strand break. But if you also could deliver a donor template, you can get homology-driven repair, and you can introduce very precise mutations. And so that's how we've been now creating all our different patient mutations. And one of the things we can do is take a fertilized egg and inject our CRISPR reagents and get our culture blastocysts, and then test for activity and recombination to find out, do we have a right mix of materials that will then produce the mutant allele. And we originally did this with several different genes, and we use tyrosinase as kind of a nice visual reporter. And one of the things we found is we get hypomorphic alleles as well. So you can get in-frame deletions. And so we end up getting a couple of hypomorphic alleles for the tyrosinase. And that's important for us because every time we go to create a new NF1 model, we're always on the lookout for an indel that will mimic a naturally occurring patient mutation. Again, there's at least 4,000 to 5,000 different NF1 mutations that have been identified. And so we can, in the process of creating a precise mutation, we often will get something that's close to what another one is. So the cDNA really was a boon for how we were doing things as well, because we could then start making predictions based on activity. And so a lot of it just happened that the sponsors, the philanthropy that was supporting the different mutations that we were making, were all different types of mutations. And I'll talk about a deletion mutation, a missense mutation, a cryptic splice site mutation. And again, we have kind of these peppered mutations all along the gene. And so we've published this. The N group has really done an outstanding job of really creating a rapid in vitro system for us to look at mutations and the functional consequences of those. We also, the CRISPR's allowed us to create cell lines, null HEC293 cells, iPS cells to create specific mutations. So that's another way for us to make NF1 deficient or patient mutations in cell lines rather quickly. So I do want to focus on a couple of mutations. Again, so we made the premature stop mutation that we had created our first mouse with. We made that mutation in a mouse cDNA and then expressed it in a cell culture line. And one of the things we noticed was that we end up getting a truncated protein in our cell culture system. And this, here's my pointer. So here's the expression of a wild type cDNA. And you can see we're really way overexpressing this. I mean, the normal control NF1 level is shown here. So we're 100-fold overexpressing this. But in this model, we had a couple of our patient-specific mutations that were producing fragmented proteins. And this is the truncated one from that first nonsense allele. And we also see elevated phosphoric activity in this particular model. And this work was done by Kurong Lee, a former postdoc in the lab. And going back to this, we were wondering, so about this time, I had actually, when we first made the mouse model, we sent it to David Gutman's group and he bred it to their GFAP model. And David called me one day and said, you know, Bob, I think this mutation is actually doing something else. That is, it's worse than a straight null allele. And you know, at first I wasn't thinking too much about that, but he said, no, it really is. So you need to go back and look at some of your data. And then once we started looking at our cDNAs, we realized that this mutation may have some dominant negative activity. And I think we may have gotten this from Frank McCormick's group last week when he's, or the last time we met, in which he described what was going on with NF1 being a dimer. But the key here is that if you do have a mutant allele that's functioning dominant negative activity and binding, you don't end up with just 50% activity. You only end up with 25% activity or even less, depending upon how stable that mutant protein may be. So I think the key here for us was that not all the mutations are equal. We had suspected that we might have some dominant negative, and we also suspect we have hypomorphic alleles, particularly when we start looking at some of the mild mutations. So what I want to do is give you an example of what this really interesting mutation at position 1466. This is a missense mutation, but it's due to a cryptic splice site. And what this mutation does, and Ludwig was again involved early on in her career with this particular mutation, identifying it, and there's been, all the mutations are rare. This is one of the more prevalent ones. By that I mean common. There's 37 cases reported worldwide, at least in the database, in the LOVD database as a couple of years ago. So in this particular case, that point mutation, that single A to G change, unfortunately, completely recapitulates the normal splicing site. And so what you end up having is this sequence is identical to the splicing site. You end up with a 62 base pair deletion. And the consequence of that is you end up missplicing, and you end up having a frame shift and the wrong amino acid substituted. You get a truncated protein with the wrong amino acids, and then it's rapidly degraded. So one of the things that was important to us is, well, there are ways of correcting cryptic splice sites, and you could use antisense oligonucleotides as a therapeutic approach for suppressing this aberrant splicing. But one of the key questions was, well, even if you're able to suppress that, the produced protein is still going to have the wrong amino acid there. So it was important for us to express with the cDNA to test out what would happen if we had the wrong amino acid in that case. And so we're there with the tryptophan 489 to cysteine. Turns out that we can make the protein, it's perfectly stable, and in fact, it functions just like wild type when we look at what happens in our cell culture model, is that it suppresses phospho ERK activity. So now we're going to, well, so we think that we proved that the cDNA may work. Deanne Wallace's group started working on making a cell line. They made iPSC cell lines that are homozygous for this particular mutation. And this is the work of Eli Awad, a postdoc in her group. And they had designed, once they had this homozygous iPS cell line, they designed some antisense oligos, the morpholinos, to block this cryptic splice siting, and then wanted to demonstrate, okay, well, if this worked, then do we get normal splicing? And that's fortunately the data that my slide will move is here. So this is the parental embryonic stem cell line showing the normal amount of neurofibromin expressed in this parental iPS cell line. Here is the homozygous null or homozygous line that has this specific mutation. If you treat with control oligonucleotide, you don't see any production of NF1. But if you treat with the morpholino, now you're seeing NF1 that you can detect by Western. And more importantly, not only detected by Western, but that's sufficient with that morpholino to produce enough neurofibromin that's at least in the homozygous iPS cells allows you to suppress the elevated GTP RAS levels in this line. So that's great. Everything appears to be working. Well, how are we going to prove this works in vivo? So in this case, shown here is Laura Lambert. Laura does all of our genetic modeling, creating our mice and rats. She does the design, and I really highlight her work with the rat model here in a few minutes. But Laura, we can't just make a simple mutation in this case for the mouse, because if you make the point mutation in the mouse gene, it doesn't have the same surrounding genetic context, and so you won't get the aberrant splicing. So in this case, we had to humanize exon 13 along with the patient mutation. So in that case, we've replaced all of exon 13, 50 base pairs of the mouse, or I'm sorry, 50 base pairs of the human flanking introns, and then now we're going into mouse sequence. So we now have this mouse, and then now we're breeding it into different genetic models, and I'll tell you something about that in a second. So another model I wanted to talk about, and this was work originated with Lou Lay and David Gutman as part of a collaboration sponsored from the Giorgio Foundation, where we really want to characterize what's going on with the mild mutations. And so one of the models, the 1809 model, I'm not going to talk much about, other than we did create that model. David's group, I think, you'll be learning more about the phenotyping on that model soon. So I'm going to focus on the other mild mutation that has been reported in patients. In this case, patients have pigmentation-related phenotypes, but really they don't develop cutaneous or plexiform tumors. So we were surprised when we made the mouse model, and this is the delta methionine, so the deletion of 3 nucleotides, the AAT, that produces a methionine 992 deletion. And we made a knock-in mouse model with this using our CRISPR technology. And so in this case, we do get, when we started doing het-by-het crosses, we ended up with, we're not getting very many animals born, so there appears to be some lethality, but we're seeing some animals that are homozygous that are surviving at a young age. And these animals are runted, so these are homozygous. Here is showing you their images of them at 6 and 10 days of age, and their body weights. And this is work that's been carried out by the undergraduate student, very talented Alexis Murowski. And if you look at the protein expression from these animals, it appears that the NF1 protein with this mutation is stable, at least from the brains of the homozygous animals, and the heterozygous and homozygous, we're not picking up much difference. By Western blot, we've got more blots to do. So the other thing I want to point out is I'm really showing an awful lot of unpublished data today, but that's more fun to get everybody up to date, and I'll be glad to share these models if anybody wants to work on them. So this is the delta methionine knock-in model, and we recently have some more information on this model. We presented some of this at the CTF meeting, the last one that was held in person, and what we saw was these animals have an abnormal skull, brain casings, and when we started removing some brains for CNS evaluations, one of the first things you notice is here's the homozygous animal here, and the whole rear of the whole skull cap is rounded off, it's shorter, and now you can see that the brain is a little bit misshapen. You can see it's pushed up into the cranial cavity. When we first took out these animals, the other thing we noticed is the pituitary appeared to be a little bit smaller. Not sure, we're still collecting additional animals to look at that now. But I think one of the most exciting things from this model is that, and we're now looking at waiting to get micro-CT data back from the entire skulls of these animals to really evaluate and get accurate measurements of what's going on with the various skull landmark features. But the most exciting data is come from our recent work in collaboration with Kai Zhao. So Kai is a faculty member in my department, and we're very lucky, he's a heart development guy. And so one of the things we notice with the animals is that the surviving pups, if you go to perfuse them, then their blood is just really dark. Particularly if you look at the descending aorta, it seems to be engorged. And so there's something going on with the cardiovasculature. And in looking at P0 animals, Kai's group has demonstrated that, in fact, what we're seeing in these surviving homozygous animals is that they have a ventricular septal defect, and that results in the double outlet phenotype. And in this way, it captures some of the Noonan-like features that have been reported with mild NF1 mutations. So I think what's going on here is that there's a problem with NF1 and the neural crests, which are neural crests, the small population. They're very important for aortic branch formation to be normal and for the development. And so I think we now have a good handle or a good model to then go after what is NF1's role with those neural crest cells and with the aberrant heart function. So let's check time, see how we're doing. Okay, so these are some of the models that we've been creating, and so I wanted to touch briefly on a couple of the new ways or genetic systems we have evaluating them. So I'm often
Bob,
asked,
can I ask a question?
Sure.
This is Peggy.
Yeah,
What happens when you put some of these mutants on different strain backgrounds? Anything with phenotype?
we haven't had the opportunity to do that at all.
Okay.
So everything that we have is on a pure B6. You might question the last mouse, why is that animal white if it's a pure B6? Those are actually tyrosinase deficient. We have an albino colony that we use for production. And it did occur to us that we need to go back and restore tyrosinase just as an added control that there's nothing going on there with that pathway. But I will tell you, I've been trying to connect pigmentation, which I know a fair amount about because of the melanocortin system, to NF1 for several years, and I can't get there. So I don't think they're connected.
Well,
okay.
What about cafe lay spots?
Well, yeah, but what I'm getting at is I can't connect the melanocortin system to the NF1 cafe LA spots. Yeah, but we are working. Deanne and I have a collaboration with someone, and Laura and I have Lambert have a collaboration with someone here at UAB who studies melanocytes and pigmentation.
Great, thanks.
Yep. So I'm often asked, well, what's the best model for testing NF1? Well, the best mouse model, if you want to look at plexiform tumors, I think probably one of the best is that desert hedgehog tree model. But it takes a long time in a lot of animals. One of the things we had noticed using, so another model that I think is very good, although it has zero relevance to what goes on in humans, is what we call an acute model. And this is a systemic loss of NF1 all at once in adult animals. And the way we do this is we use a CAG-3 to express a tamoxifen-inducible version of CRE throughout the body. Combine that with a flox allele, and now when you inject these animals with tamoxifen, we give low doses for 5 days. What you see is that all the animals die within about 12 days. I mean, this is a highly penetrant phenotype. And what happens here is animals waste away, even though they're eating surprisingly normally, but they burn through their adipose tissue and they even burn through some lean mass. And so and we have a manuscript and draft that's for this phenotype. And we have a lot more detailed analysis on what's going on with energy expenditure in this particular model. But the key here is that with this mouse, it is artificial and you're turning off NF1. There's still some residual neurofibroma you can detect by looking at brains and Western blots. But in the presence of tamoxifen, we really knock down NF1. Whereas if you don't give you tamoxifen, these animals are just fine. So this is probably one of our quickest models. And in fact, we're employing this to now look at some of our NF1 patient mutations and indeed the Nick Gilbert's mutation at which we created that model. So the other system that we have been developing in the last few years is a GABAergic deletion of NF1. And when I first started collaborating with David Gutman, David had mentioned me. He said, well, Bob, I have this mouse that is extremely obese, and I don't know what to do with it. And I don't think David knew that I worked on obesity at the time, other than having a personal interest in it. And so I said, well, Dave, I'll be glad to take that model. And so he had originally used a flox allele with exons 31 and 32, and using a GABAergic knockout. I was really interested in trying to recapitulate this with a different NF1 flox allele, because I've studied a lot of obese mice over the last 20 years, and a lot of times the genetic models don't behave themselves and they don't really reproduce. But we've now seen the same phenotype in two different systems of flox alleles for NF1. And the key here, and this is all work by Kahoot recently, is that if you breed and produce animals that have a GABAergic knockout of NF1, shown here is the weight curves or growth curves for females. You see that these females get obese really quickly, and see these animals are extremely hyperphagic. Males also get obese, not quite as fast as the females, but they'll also get fairly obese, and they'll get up to be around 55, 60 grams. And so early on, they're really a model of hyperphagia that's really similar to a melanocortin or a leptin deficient animal. But really they are an animal model of diabetes. And so if you look at their water intake for these particular animals, at a very young age, they really are taking in huge amounts of water. And I'll show you a picture of the pancreas in a minute. So these animals, they're hypermetabolic. If you look at their heat production by indirect calorimetry, you can also see increases of the knockout over the controls. And also they show increased activity. And this is when they're still young. So if you measure them around 8 to 10 weeks of age, when they're in that growth spurt, then they're hyperactive and hypermetabolic. If you wait till they age out and they're obese, they start slowing down. They're actually less movement than other controls, but that's to be expected for a really obese animal model. The interesting phenotype here is that these animals are showing this activity and increased activity and hyper and heat production just before lights out. Just before lights on. So this is the dark phase, and this would be light phase here. And so these animals get very active, and you see they even kind of stay active. I didn't show you the data, but we also noticed that these animals don't, they appear to grind their food, and they eat throughout daytime and nighttime. So normally animals eat 90% of their food during the night, or mice do, but these guys are eating throughout the day. And so that's what's really driving the hyperphagia.
Hey, Bob, quick question.
Sure.
So they're hyper metabolic, but they're getting fat too?
Yes, they are eating that much.
Oh, I see. So it's because of the hyperphagia, they're still taking in too much. And these are the homozygous mice, right?
So they're flox, and
right.
they're just, so they would be so it would be both copies are deleted in GABAergic cells.
Right, but there's nothing in the heterozygous mice.
No, nothing in the heads are fine.
Okay, thanks.
Yeah, or at least with the metabolic phenotype. Actually, there's a previous publication by David's group and others looking at GABAergic heterozygous animals, and so maybe we can talk about that later. So these animals are diabetic, they're hyperglycemic, they're hyperinsulinemic. If you look at a glucose tolerance test, they're also the area under the curve. So the DLX knockout really are a model of diabetes. And in fact, I mean, here you can look at water bottles. You can tell which animals are this is early on before we stuck them into our automated cage system. But these are the GABAergic knockouts, and within a couple of days, they'll drain their water bottles. When we looked at the pancreas, and we got a hint early on when we did this first staining, what you can see is that the beta cells appear to be relatively normal. So you do see some hypertrophy of the pancreatic beta cells. So they are, that goes along with the hyperinsulinemia. But really the glucagon, so the alpha cells, so here's a wild type animal over here. You see this nice ring of alpha cells around the islet. Here you see a complete dispersal and disruption of the islet cells. And so we're still trying to figure out, is the glucagon axis playing a more of a component here with these animals? And we're trying to follow up on that. There's a fair amount of literature about GABAergic signaling within the pancreas. And so one of our questions was, well, did we knock out, is there a role for NF1 in the pancreas? And so we went back and using an MTMG reporter, we see no detectable recombination within the pancreas itself. So we don't think it's NF1 in the pancreas that's driving this phenotype. We think it's really innervation of the pancreas is more likely what's going on. And in fact, now we're looking at some EM work to look at the nerves innervating the pancreas. The other thing that we noticed was a behavior.
Don't. Sorry, just wanted to let you know that you could try to finish up in about next 5 minutes or something, just to leave some time for discussion.
Yeah. Sorry about that. Yeah, I knew I had a little too much. So quickly go through a couple of things. So these guys have aberrant nest building behavior. So we think this is also a potentially good behavioral model. I do want to talk about our rat models real quickly. Laura Lambert developed these these heterozygous rats. And the reason this is important is heterozygous mice don't have much of a phenotype. The heterozygous rats have a very robust phenotype. And so we had made this missense mutation that again is associated with spinal NF, different missense mutation. In the process of doing that, so we made the point mutation. The heterozygous animals, we also got an indel that provides a nice knockout allele. So we have both a knockout allele and a knock-in allele. And what we see with our knockout alleles, so knockout animals, females, these plotting percent of animals that are tumor-free, and all the females have that are heterozygous for the knockout allele develop tumors very rapidly. This goes along with work that Matt Steensma published, and we had collaborated, helped him out a little bit on a few things. This completely recapitulates what Matt's group had published. The interesting thing was this knock-in allele, in which here the females do not show tumors, or very few of them do. However, the moment you mate these animals, boom, you get tumors in either the knockout or knock-in. So there appears to be a hormone responsiveness or role in these particular rat models. Shown here are a couple of the rats with these tumors. We also looked at the spinal cord carefully and see that some of the males are developing some aberrant signaling in the spinal cord. These are HER2, ER, and PR positive tumors, or the triple positive tumors that we see. And probably one of the most coolest things we're seeing right now is that these mammary tumors in these heterozygous rats, again, these are heterozygous, that you see metastases to the lungs. And so now you can see the lung contribution here. Both these lungs have been hit with tumors originating from the mammary gland. And Kelly and my group is, along with Alex, helping out a few things, has done some staining. And so now we can look at lung tissue and see mammary specific markers. So we know that these are metastases to the lung. The other thing is that these point mutation is actually driving other NF1-like phenotypes. So our male rats are getting gliomas. We have a male rat with chromocytoma. We have some that have bleeds within the spinal cord. We also have these lymphoid nodules in the colon. And this is another one of those rare things that's associated with NF1. So we think that this rat really recapitulates much of NF1. Virtually no behavioral phenotype. I showed this to a previous NF1 meeting. We're really disappointed that they don't have a CNS phenotype, but we do see knockout have reduced protein in the brain. HETs are somewhere in between. Phosphoric levels appear to be relatively normal. We looked carefully at the optic nerves, measured optic chiasm, and so those all appeared normal. And so I think what I'll do is just kind of end up by saying that we're applying these models and doing, as part of our way of delivering NF1 cDNA with nanoparticles to try to rescue phenotypes, and then also to create nanoparticles that will deliver CRISPR-Cas reagents to try to correct these mutant alleles. And then I think the real challenge, we'll skip all the mutations that we're doing. So the real challenges that we have is, you know, how do you optimize the route of injection? How do you deliver these reagents such that you can get correction of the mutant alleles and really have a robust therapeutic response? And I think that's really key for us going closer, or as we get closer to personalized medicine, is how do we really make use of these systems to create and test better therapeutics? So I realize I rushed through that last little bit. Here's an acknowledgement of all the people that have really done all the work. Laura Lambert and particularly Deanne Wallace's lab have done a majority of you know, helping to create these model systems and doing a lot of the analyses. I pointed out the people that did a lot of the work. And then lastly, Jim pointed out that we have a new U54 at UAB grant for precision animal modeling. And I'll say that here's a slide of the leadership of that group. And one of the things you'll see is a lot of NF1 people here. You got Bruce, Laura and I, and Deanne. And so the reviewers really liked what we had done with personalized medicine as part of this application to become a center. And I think other diseases are going to start taking this approach. And I think we were kind of a vanguard of applying this. And we've been very fortunate with a lot of the groups that have been funding us, particularly the Gilbert family. And I mentioned the Giorgio Children's Tumor Foundation, Nothing is Forever, and the support from Bruce. So I'll stop now and take your questions.
Thank you very much, Bob. That was great. So open the floor to any questions that anyone has.
I have a quick question. So,
Hi,
hi, Bob. This is Uli. Thanks for a great talk. I'm wondering. So most of your models have something like a yes or no answer. You have one allele with a mutation or not. And my question is, do you have any data that help us understand if, let's say, only 5% of one allele or 10% or 15 or 20% of an allele has the correct sequence, how does it help the disease phenotype?
So I guess what you're asking is, can we model mosaic NF1 or forms of mosaic NF1 where you're
so not mosaic as part of cells. So basically within one cell, you have a mixture of mutated and non-mutated one. You already have correct and incorrect alleles, right? So for example, do you know whether the expression level between the mutated and non-mutated NF1, to what extent that influences the phenotype?
So unfortunately, no, we do not have that. So one of the things that one of our side projects is we've been attempting to insert halo tags and other tags to the NF1, different NF1 alleles, so that we can get a better handle on tracing the activity of a given allele, and also a way of tracing the protein as well. I think we're very interested in knowing where the mutant protein's going, and you know, what is it that they're not doing. I'm fascinated by, so everyone knows that NF1 regulates RAS. I think the new data coming out of Eric Chain's group at Baylor, showing that NF1 is a co-repressor of the estrogen receptor signaling pathway, directly interacts with estrogen receptor. That's fascinating as it's a new role for NF1. So I think in the end, we will hopefully be able to classify, much like they've done for for the CFTR, cystic fibrosis. So different types of mutations, where are they getting hung up? Are they not processed correctly? Could you use correctors to stabilize proteins? Can you give drugs that will help chaperone certain mutations to the right place? Can you come up with estrogen-specific compounds or compounds that will work on NF1 to either increase or decrease roles with estrogen receptors? So I think we have an awful lot to learn about this large protein that's highly conserved across all species. So
yeah, thank you.
Hi, can I jump in with a question?
Please.
Yeah, it's Patrick Harrison. I work on gene editing for CF, but I'm really interested in this. Just, I think you mentioned you had one model which you could potentially treat with antisense oligos.
Yes.
And I'm just wondering, could you get some clues from that about which organs you can maybe treat or which symptoms you can specifically treat, so that then you could focus on that with the delivery route with the CRISPR options or something like this? I'm just thinking you might get a lead with that first, which you, if you're trying to get to clinic, the antisense might be the quickest, but it may not be the most versatile. But if you could find a delivery route, that might give you some head start.
Right. So I think that's a great idea. And I think, again, that was one of my last slide. I think that's the real challenge is, well, we have several hurdles, but if you can find a therapy that works, that's step one. Step two, how do you deliver it to the right place and at the right time? I think we have to also be wary, particularly of CNS effects. Are you going to be able to treat children or adolescents with cognitive issues with any sort of therapy, and where the brain is probably already formed and made certain connections, and you don't know that you'll be able to reverse certain things. So timing is everything. I like your idea about maybe using. I hadn't really thought about using that model as a way for delivery system, but because it's antisense oligo approach, that actually is it's a great idea, because that could be delivered a number of different ways. Yeah.
Thank you.
No, that's great. Sorry about rushing, guys. Again, I like to talk about a lot of new data that's not published, and so it's hard to kind of gauge am I, going to hit the mark or not.
Oh, that's okay, Bob. I'm sorry I had to rush you at the end.
That's all right.
Does anybody have any other questions before we?
Can I jump in with another one?
Sure.
Go ahead.
I know Dave Bedwell's on the call as well, and I'm fascinated by the read-through idea. All of your read-through mutations, or PTC mutations, sorry, are in early exons. Is there a PTC mutation in the last exon, which you could maybe test some of these compounds in to maybe get round or dissociate the nonsense-mediated decay from the read-through? Do you have a model for that? Would that be of any use?
So the answer is we are working on models. We are creating a couple of nonsense some exon skipping as an approach that Deanna Wallace is very interested in, and we have proof of principle that we can skip certain exons, and also within Andre Lear's group. So for his so I'm collaborating with Andre in a ribozyme approach, and so we have created some mutations in, I believe, in exon 52 and 54, and in the process, I think we created a nonsense allele there. We haven't expanded those to use them for that sort of approach. I think David will tell you that we kind of handpicked a few of the nonsense alleles to produce, because again, we already know that nonsense alleles don't behave the same way, but some we know up front, I think, are going to behave better than others. And I think David could maybe elaborate on that.
Yeah, so obviously we don't want to work on the really hard ones. We want to work on the easiest ones first. And then if we can get something that works there, then we can transition to the harder ones. Now, as far as NMD goes, I agree that if we were at the last exon, that should largely eliminate NMD. However, what we've been finding is that if we can get enough read through, we can increase mRNA abundance spontaneously, at least partially. So we don't think, it depends on how much NF1 you really need to produce to get a phenotypic benefit, but we're hoping that by just getting enough read through, we can, get kind of a feed forward loop and try to get enough mRNA to amplify that read through effect and get enough NF1 to do something.
Yeah, okay, fair enough.
Hi, Bob. Can I ask you a question? A very nice presentation. Beautiful. It's George Dixon here from London. Bob, I'm curious that in the rat, you see quite a different phenotype, or more severe phenotype than in the mouse. have you looked at all at strain background in the mouse, whether the strain background produces a difference in phenotype in these animals? It feels like there's some sort of modifier effects here that really aren't clear.
Right, and I think that's a good point. All of our mice are on a black 6 background. We do that for the ease of genetics and creating the model. We wish we had a whole lot more money where we could go back and create some of these lines or outbreed them. We have, we've talked about it in our lab meetings. I don't know if anyone's actually started it yet. The interesting thing about our rat, it is an outbred rat model. So that for me is really surprising. I would expect more divergence in the phenotype that we're seeing in the rat or some divergence in the penetrance that we're seeing. But at least with the mammary tumors, it's highly penetrant. And this is an outbred strain. And even with the point mutation, the spinal mutation and it's at the tip of the arginine finger, that we're fairly sure that that's interacting or disrupting RAS signaling. That model, we were expecting a little bit more divergence in the phenotype, and maybe we should be happy with it. We are getting, we are recapitulating what's seen in humans more than in the mouse. And all I can say is, a mouse is not a rat, and I've made my career in building genetically modified mice for the last couple of decades. But I'm beginning to suspect that for NF1, the rat may be the better rodent model to study.
Okay, thank you. Thank you.
All right, thank you, Bob, very much for a great talk. So if anybody has any additional questions, maybe you can direct them to Bob on his email or contact him. So thanks everybody for attending this particular seminar. So we are in the midst of setting up the next seminar. It will likely be in May, 3rd Wednesday of May. We're not exactly sure yet, but we will keep everybody informed. So everybody have a good rest of your St. Patty's Day and enjoy. Thank you very much. Bye.
Thanks, guys. All right, bye-bye.
You guys, good talk, Bob.
Thank you.